This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1141, recorded on August 12, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent, and everybody else. Looking out the window, it is a largely overcast day. I have not been outside, believe it or not, so I don't know what the weather is like, except that it looks not too bad. It's probably in the low 80s, humidity around 55%. That's my guess.
Very good. It's 50%. You're really good with that.
Well, okay then.
It's 27°C here and cloudy. But this morning it was
I have
very
nothing
sunny.
more to say.
It was sunny this morning. Very nice. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. 96 degrees
Yes,
Fahrenheit.
of course.
Sunny. It's Texas.
It's Texas.
I don't think it's going to be 100 today, and if it's not 100, that's a good day.
So, Rich, all those people that were without power for a long time and we kept hearing about, do they now have their power in Texas?
As far as I know, but
Oh, okay.
that's some distance from me. That storm didn't touch us.
Right.
So I haven't heard anything recently, and I would say no news is good news.
Indeed.
And from Madison, New Jersey, Brianne Barker.
Hi, great to see you all. Surprising to no one, it is also 80 degrees and overcast here, just like others have reported.
Right.
We are recording on a Monday because, well, we tried on Friday, but the power stopped here in New Jersey anyway. And it did not resume in time. So we're doing it
Powers
again.
that be were not.
Yeah, we're dependent on technology, right?
Mm-hmm.
We are.
In all ways.
We are.
I'm thinking back to the hunter-gatherer era, you know. We're dependent on food and a shelter. That's about it, right?
Now
I
see
saw
why people-
I saw a news bit last night about space junk.
Oh,
I know
me
you
too. I
watch
saw that
PBS
same thing.
You
Yeah, I
watch
saw that.
PBS NewsHour, Dixon.
Yeah,
I
I
know
saw
this,
that
so
too.
maybe
Yeah,
you saw
yeah,
this.
yeah,
And
yeah.
basically one of the things there is how dependent we are on stuff that's in space,
Mm-hmm.
and how if space junk gets sufficiently out of control so that a lot of the space stuff that we depend on goes down, we got a problem. You can't use your credit card, right?
That's right. Or you won't know where you are.
Or you won't know where you are. Of course,
Yeah,
I don't know
that's
where I
right.
am anyway. So
Oh, my gosh. But there's that movie, what was it, Gravity, where the
Yeah.
space junk...
yeah.
People have said that's highly unlikely that happens.
If you
Well-
look at the amount of space between those objects, you'll realize why they say that. I mean, they make it look like they're all going to crash into each other when they show you the Earth, but the Earth is very large.
Mm-hmm.
space
heard-
is big too.
When you expand, and like when they show you how many airplanes are in the air, I would swear that they're all going to crash into each other, but they don't even see each other. There's so much room between them. So it's all relative to the way you present the data, right?
Mm-hmm.
And that's how they presented it, like it was an emergency. And it probably is, but they're still releasing satellites by the hundreds.
I know.
Hundreds.
By the hundreds. They'll send a satellite, a rocket ship up, and it will release 100 satellites.
Yeah.
And each one has a little job to do, and then I don't know what happens to them afterwards, but
They
they'll
become
get
space
together and have
junk.
a party, and then and then they wonder, like the astronauts on the International Space Station, when will I be going home?
Mm-hmm.
And they don't know either now. They have no idea of
Why?
when they're going to go home.
They're negotiating with a different
Yeah, I
airline.
know that. That's bad. That's
With carriers?
definitely a bad move.
Yeah, they may come home on a dragon.
Lord.
If you
Yeah, I
like
know.
these discussions here on TWiV and all our other podcasts, we'd love to have your financial support for them to continue. Microbe TV is a 501c3 nonprofit entity, which means if you're in the U.S., any monies you donate are federal U.S. tax deductible. So you can go to microbe.tv/contribute. We have a number of ways that you can help us continue science communicating. I hope you all listen to, not the hosts here, but the listeners. I hope you listen to Long COVID with Judith Brookfeld. That was an episode I recorded in Stockholm back in May. It was really good. Short and sweet.
Hey, Vincent.
Yeah.
You need to reach out to one of your moderators because they sent me Alex Trebek forever stamps to my office, and I'm very excited about them.
That's nice.
This is awesome. So thank you, moderator, for this.
That was Les.
Thanks, Les.
Les from California. He's very excited to send them to you, Brianne.
Well, I'm very excited to receive them. So excellent.
Okay, some news before we get to the literature. Very interesting. Here is an article in CIDRAP. WHO to convene emergency committee to weigh Mpox spread in Africa. Now, this is from August 7th because the DRC has had an outbreak since 2022, 14,000 cases, 511 deaths. And this is from a novel clade 1b virus, which is thought to be more lethal, I think. Right, right, Rich?
I think it has intermediate between the really deadly one that has a 10% case fatality rate and the one that is not so bad, that at least had a 1% mortality rate. That was before all of this outbreak stuff happened. Those were the case fatality rates, and we always felt lucky that it was the less lethal one that got out and was circulating. And in fact, turned out for reasons that I don't understand, probably having to do with the populations that it was in and the quality of the healthcare and the vigilant, turns out to have a lower case fatality rate than 1%. And I think this one is sort of intermediate. Aside from that, I have not, although I haven't looked at it in detail, I see nothing about this that indicates there's anything else special about this clade that makes this different than what has been going on.
Can you refresh me? I'm sorry to interrupt. I'm sorry.
Go ahead. Go.
I'm just curious. I forget where all of this comes from. Is there an origin for the monkeypox, the Mpox?
It's African, and there is probably there seem to be foci in the DRC and in West Africa, and the clades have different foci themselves. I don't have the details on the top of my head, but that has been the focus of monkeypox for as long as we have known about monkeypox, which, and wait a minute, I forget. The disease we're calling Mpox, and the virus is still monkeypox, right?
Correct.
Okay.
And there's some reservoir of some maybe rodents or other small mammals in that portion of Africa.
Yes. The thinking is that it resides in a rodent reservoir of some sort, and the infections of humans start at least as spillovers, but there can be human-to-human transmission. The outbreak that happened in, was it 2022, that spread through Europe and the U.S. and the Western Hemisphere was unusual in that it was spilling out of the of Africa, and it was primarily sexual transmission, and the population most seriously affected was gay men, so transmitted sexually. I have seen nothing in this report that indicates that there's anything substantially different about the transmission that we're talking about here. So it remains a mystery to me why this is persisting as
is there any data as to how long it's been around?
Oh, probably. I don't know about data, okay, but probably for a long time.
So where's the balanced polymorphism is what I'm driving at. Why hasn't this resulted in a series of immune human populations that are not susceptible or less susceptible to the
Well, you know
pathological
that
effects?
you raise an interesting question there, because I think monkeypox has probably been around forever. We did go through a period of time where the entire globe was vaccinated against smallpox.
Right.
All right, and that vaccination covers monkeypox, and
I
there was a
see.
reemergence of monkeypox in Africa, coincident with the waning of immunity from smallpox. Not really waning immunity, but the introduction into the population of people who weren't vaccinated.
Yeah,
Okay?
maybe the low mortality rate would not result in a balanced polymorphism like it does for malaria, which has a higher
I
rate
see
of
what you mean.
mortality. Yeah.
I see what you mean. This does not spread easily. Okay.
Right.
And it requires really, very close contact. At least the history so far is that. So there's a not a large fraction of the population that is effective, and there's been, up until the last couple of years, the outbreaks have been actually quite small. But then there was this spillover into Europe, and this continuing problem in the DRC. That's a lot of cases that now have people sufficiently concerned that, are they going to declare this a PHEC? What is that?
Public health emergency of
concern.
Well, international would be a pandemic, so of concern, I guess.
I in the other article that I think we'll talk about, there's discussion of making it a continent of concern or continental issue of concern, which I'd never heard of before this.
So WHO wants to mobilize funds and get vaccines. So there are 2 vaccines, and they have to be emergency use approved in these countries and have to be paid for. And so Japan, US, EU are all working on donations, which is good.
And the other, I mean, we know a lot about these viruses. Okay, so the preventative measures are good. We know enough about the transmission of this so that, in fact, what I think stifled the epidemic, the global epidemic some time ago, was education. Okay, people learned how it was transmitted and learned to avoid behaviors that would transmit it, learned to recognize the disease and take appropriate precautions when they had it. And so it's the kind of thing that those kinds of measures can help out with. And I was going to say, there's also a couple of very good antivirals that work both on smallpox and on monkeypox. So we're situated pretty well here.
So, Rich, I have a question. Do you think that before widespread smallpox vaccination, people were potentially always distinguishing monkeypox and smallpox, or do you think that that maybe monkeypox was more of a problem than we know of in the past, and that vaccination sort of reduced that? How different are the clinical
I can
symptoms?
only speculate. I have no data. What jumps to mind is that smallpox was probably a much bigger problem than monkeypox, to the point where smallpox itself may have conferred a population immunity that kept monkeypox tamped down. Okay. Beyond that, I could imagine that if you got a case of monkeypox that went away, you might very well think you dodged a smallpox bullet. You didn't even know it was monkeypox,
right?
But because I think that symptomatically the diseases are pretty much if you look into a person's history with their symptoms, you might be able to tell that it was a contact infection, whereas smallpox is a respiratory infection that starts with a bunch of other associated symptoms. But I wouldn't be at all surprised if this could be easily confused with smallpox in an environment where smallpox is circulating. And Africa, of course, was, a trouble spot for smallpox.
So if the rodent is the reservoir host or the natural host, as perhaps it is, then how does it jump to people?
By contact, okay? Bushmeat, Okay.
So you should stop
Or-
having sex with rodents. That's it.
Well, they come in your
you
homes,
should-
Dixon.
Yeah. You
Well,
should.
no, of course,
You
but
should not.
It's the
you sh-
urine that transmits it.
You should not. Okay. So I think it's not very frequently that the transmission is actually directly from a rodent to a human. It's called monkeypox because primates, other primates, can get it, and I think stuff like bushmeat can be a problem. Okay?
I think there are some situations where there may be some bushmeat with rodents as well, unfortunately.
Okay, fine.
So we have an article in Science by John Cohen. Africa poised to declare continent-wide emergency alarm for Mpox outbreak. Rollout of vaccines will likely be a challenge as 16 countries fight active outbreaks. So there's a little more detail on what they're thinking of doing. PHECS, Public Health Emergency of Continental Security, as Brianne said.
Yeah, I've never heard of that
It's
before.
a new one.
Probably a new one, yeah.
A lot of cases. So for those of you who think this is not a thing, think twice. There's a lot of Mpox. Okay. This is very interesting now. Well, it's all interesting, folks, right? But this one is pretty cool. CDC has just published mortality in the U.S. provisional data for 2023. It's fascinating. So 2023, how many people do you think died in the U.S. in 2023, Dixon?
A lot.
100,000?
No, more.
Nah.
Many more.
Gotta
3
be
million,
millions.
just over 3 million.
Yeah. That would be
And
my
what do you
guess.
think the leading causes of death were?
Heart disease,
Got it.
stroke, cancer.
Cancer is some number 2. And number 3 is really very interesting. Unintentional injury.
Oh, you mean like getting shot?
Well, you forget that there's a step and you fall into the gully or something.
Oh,
just
that
like-
I see.
or you don't look twice when you cross the street.
Or car accidents.
Car accidents, all kinds of
Okay.
stuff.
Okay. How about homicide? How far down the list is that?
And this is a good question.
It's not listed. I wonder if that is also an unintentional injury.
Really? I know, quite intentional.
It's a
actually.
manslaughter or not, Homicide is not unintentional.
No.
Anyway, the interesting thing here is that the number of deaths from COVID-19 in 2023 was 76,446. It's about 70% lower than in the previous year. So it's still a lot of deaths, right? That is big. It's bigger than most influenza seasons.
Yeah, I mean, it's still the only, individual microbe that is showing up on the list.
Yep.
How many people actually had COVID?
Mm.
What is the death rate?
Well, I would guess at least 100-fold more because the death rate is probably 1% or slightly less.
I mean, in the beginning, it was really.
Yeah, well,
conceived
the
to be
beginning
very
was
high.
it was not. Yeah, it was hard to tell because we didn't know what to do. But
Yeah, I'm
Right.
not sure that we have great data on that right now, Dixon, because if we think about it,
yeah,
so many people might be testing positive, say at home on a Binax test
All
or
true.
something like that, and then
Yeah,
not
no,
ever
you're right.
getting recorded.
That's right.
yeah, that's exactly right. We're not reporting test results anymore, so who knows?
Hmm.
But I mean, you just
Boy, the contrast between 1922 and 1923 is dramatic.
2022.
2022.
Yeah, that's right.
2022 and 2023
It is dramatic. Yes.
So
Ah.
it must mean that people are learning more about how to treat acute infections and
I-
saving their lives.
I presume that we have learned. Yeah.
So
there
that might
there
figure
also
into
might be
this.
something about that whole vaccine thing.
That whole vaccine thing.
I mean, somebody, this might tell us something about vaccines. I don't know quite what it might be.
Well, that's what I
Andre.
was also going to ask. Has the rate of vaccination gone up?
Well, we're
Despite
we're pretty much maxed out, right?
Pretty maxed out.
I don't know what the number is, but it's hard to tell because people are infected and vaccinated, right? So
Yeah.
there's pretty high population immunity, but people are dying. Why? Because they're
Why?
old and they have other issues. Even if they're not old, they have other issues that predispose them to severe disease. There's 186,500 deaths in 2022. So this is substantial.
I've
right?
had COVID 3 times and I'm old. But I've also been vaccinated.
Did you ever end up in the hospital, Dixon?
No.
It's because you're vaccinated, right?
And I took Paxlovid.
But you're also robust.
I am. That's true. I forgot that.
You took Paxlovid each time, right?
No, I've taken it twice. That is exactly right.
I mean, Daniel will tell you that when elderly people die, they're not given Paxlovid early enough.
No, that's right.
They're brought into the ICU and then
That's
it's
right.
going to have Paxlovid. No, it's a bit late for that.
Right. It's a bit late, period.
But
Yeah,
it's really remarkable when you think of what this virus has done, right?
Right.
And it's random, folks.
Yeah,
Nobody made it. It's just random.
that's right.
Amazing.
Nature did that.
That we now have this new respiratory virus that's here to stay.
Oh.
And it's going to infect a lot of people every year. It's going to kill quite a few people every year. It's to me, it's amazing, right?
We're losing our institutional memory. All of us old folks, that's what we are, the institutional memory.
So the average number of deaths, this is all in the U.S., right? From flu
This is U.S., yeah.
Is what? Anywhere from 10 to 50,000, depending on the year. Is that
That's
a
flu.
fair
That's
statement?
influenza.
Flu.
That's right.
Yeah,
Yeah,
that's right. That's right.
And so, as you pointed out previously, at 76,000, this exceeds even the worst flu year. So
Right.
it's still a significant issue.
Yep, it is. So we were here to see the onset of this,
We were.
right?
We are.
I mean, and the impact is huge because if you think, all right, let's go back to 1968, H3N2 virus emerges, right? It didn't have the impact that this did, and it's still around, but it doesn't really have the same impact. But if it's an influenza virus, it's just, well, yeah, we know influenza, but this is a new virus, and it's here to stay. This has happened many times throughout history, but we haven't seen it like this. That's the amazing part.
Come on, right? We've seen AIDS emerge.
Yeah, well, AIDS, we weren't there in the 20s to see it happening, right? We backtracked and said, yeah, probably the 20s it emerged, but this one was right there in front of our eyes. And not only that, there was a podcast that talked about it every week.
That's true. That is,
about 350 times, as I recall.
That's right.
And there's a permanent record of this emergence that will always be there. That's really cool.
If you go back to the very beginning, you can hear all the mistakes that we made.
Yeah, for sure.
Okay,
and everybody else's too.
And hear us saying, "Oh, we got that wrong,"
but
and then figuring it out.
The one episode of, I wonder if this is going to be a thing.
Yeah, right. People who know me and have listened in the past constantly remind me that in the beginning I was kind of this will blow
Of course.
over. Okay.
But we did get some things right, like saying vaccines
Oh yeah, we got a lot of
prevent
stuff right.
disease, not infection, right? Look
Actually, we got
in my
everything
hand.
right because when
Nice.
we were wrong, we said we were wrong.
Right.
I like to play with this. This is a glass coronavirus from Mark Martin,
Nice.
and I just play with it. I'm going to drop it one of these days. I really need to put it down.
You do.
But you like to. All right, we have an article from WHO. Regional health ministers rally to support immediate steps to stop polio from paralyzing children in the Gaza Strip. So this is from back in July, and as you know, they have severe sanitation issues in Gaza, and there's poliovirus circulating. So there's an effort underway to make sure that there have been no paralytic cases so far, I believe, but the virus is circulating. It's just a matter of time. So they want to go in and impose some, they want to vaccinate kids, but also do some sanitation and help, stem the circulation of virus, which is mainly in sewage.
What a mess.
It's a mess.
The whole healthcare infrastructure there is such a disaster that
It's all gone.
I can't imagine. I mean, they can't get basic necessities.
Yeah.
What a mess.
And it continues.
And finally, we have a Nature article. molecular mimicry and multisystem inflammatory syndrome in children. Remember, MISC is a severe post-infectious sequela of SARS-CoV-2 infection. And we actually had two people on to talk about MISC who found similar things as reported in this paper, which is from Joe DeRisi. Basically, when the kids make antibodies to the nucleocapsid, and some of those antibodies cross-react with a host protein called SNX8, and that is thought to give rise to the multi-system inflammatory syndrome. Isn't that interesting?
Yep.
Yeah, absolutely. Also interesting in sort of how it's meant mostly kids.
Right. Biology is very fragile. It's amazing it works.
I was going to say exactly the same thing.
There's so many
Took the
things
words
happening
right out
at
of my
once.
Well, the other thing is that it's very similar in two completely different systems. Where'd that come from? I was amazed that in the old days that bacterial endocarditis could lead to autoimmune disease to some part of your heart muscle that had nothing to do with the bacteria that was causing it. And
Yep.
how the hell does that work?
Well, there's only
Dixon
so
did-
many amino acids you can
Exactly.
use.
There's only
Well,
so many.
no, of course, if we know how it works.
Only so many ways you can arrange amino acids, right?
That's right. So that means that nature doesn't discard anything.
Overall, it's not a bad system.
Yeah, that's right. That's right.
Actually, it's too good. There's too many damn people.
Not really, Rich. That's not the way to put that.
I don't know.
We can't take care of the ones that we wish we could take care
Well,
of.
it's going to be a self-limiting proposition, right? You're going to get to a point where you can't accommodate anymore. And then,
Right.
But as Dixon would say, parasites keep populations in check, right?
They do. You just talked about it
When?
about 20 seconds ago when you were talking about another parasite.
Yeah, of course. But, they...
Infectious diseases. Let's put it that
yes,
way.
but they don't... They take away small percentages of the population, not 10, 20, 30, 40%, right?
No, but if you go back, well, yeah. No, I'm not going to start
Okay.
that argument.
Finally, that's it. So those are our news items for today. We have two papers, which are pretty cool. And the first one, which is, ostensibly labeled a snippet. But
I don't pay any attention to that anymore.
Yeah, I don't pay any attention to that. It's just a paper, and it's cool. This is in Nature Communications. The neonatal FC receptor is a pan-arterivirus receptor.
See, nature abhors a vacuum, and it just says, "Oh, I'll use that."
Of course, things get used over and over again.
Exactly.
Dixon, can you name one thing in your car that's used twice? Two different things?
Two different things that are used in my car?
Yeah, I'm just throwing something out there.
I have a hybrid car. I don't know.
Well, a tire is used for propulsion and for turning, for example, right?
Oh, I see what you're saying. Okay.
It used to be that the cigarette lighter was used for both lighting cigarettes and for
electricity.
powering your iPhone.
That's right.
Very good. Anyway, this is from a group at the University of Wisconsin, Madison, Ohio State University, Stanford University, University of Illinois, WashU, and the last two authors are Cody Warren and Adam Bailey. Adam Bailey I know because he invited me years ago to visit Wisconsin, and we did a TWiV there. And what is an arterivirus? Well, these are kind of coronavirus-like, right? It's so the arteriviridae is a family in the order Nidovirales, which includes the coronaviridae. These are plus-stranded RNA viruses with envelopes, big genomes. And the arteriviruses, well, we don't hear much about them because they mostly cause infections of non-human animals, but they're really important. They're really, they cause a lot of different kinds of diseases. They can persist for long periods of time. They're really good at evading immunity. And they often jump hosts, animal hosts, from one species to another. For example, simian hemorrhagic fever virus and porcine reproductive and respiratory syndrome virus are examples of viruses crossing hosts. So, for example, SHFV, discovered in 1964, there was an outbreak in macaques, and subsequently found in monkeys throughout Africa. In the 80s, two lineages of PRRSV emerged in swine herds in Europe and North America. They transmit really well across the placenta, so they have a big effect on fetuses. You get these abortion storms when the viruses are there, and some variants recently have emerged that are quite lethal. We don't know where these viruses came from. So these are some viruses we're going to talk about today. They're envelope viruses. They have, listen to this, 7 membrane glycoproteins. Because they think they're herpes viruses. Right? How many do herpes? Many. Herpes have so many glycoproteins.
Or pox viruses.
Pox have a lot
Pox
too.
has this entry fusion complex that's got like 20 proteins in it. That's what I was thinking of as I read this.
So they all of these, so there's 7 membrane glycoproteins, and then there's some minor ones, and so there are 11 in all, and apparently they're all needed for infectivity. And none of them look like the known proteins like class 1, 2, 3, 4, etc. They're all novel, which is amazing. So this paper is all about what allows these viruses to enter cells. And so PRRSV infects macrophages in animals, and it requires remember, viruses need to bind receptors to get into cells. So the infection of macrophages by PRRSV requires a protein called CD163. Because you can make pigs, and this is the funniest thing. I was at an event for Ian Lipkin a couple months ago, and I was in the bathroom, and there's a guy next to me. I said, "Oh, I know you." He said, "Yeah, I made the CD163 knockout pigs." I said, "This is so cool." And those pigs, now why would you do that? Because then they don't get infected. He was very proud of it.
Yeah.
He said, "We should get you on TWiV to talk about this sometime." Because now you can grow pigs that don't have CD1, and they won't get infected with PRRSV, right? That's a good strategy. But I don't know if pigs nowadays, if you go to the store and buy pork, I don't know if it has CD163 or not.
Probably does.
You could probably ask the checkout guy.
Sure,
Does this
they'd
have
know.
CD163?
I can just see, there's some advertisement for our pigs are CD for free.
CD163 fee.
I just feel like transgenic pigs would be kind of still expensive for your bacon.
So, yeah. Well, eventually you could ramp up production, right? Anyway, so the idea is that there are probably other proteins needed for entry. So that's what this paper is about. Okay. So there's some cool tech here, which we'll talk about. So first of all, there's a cell line, MA104. I forgot where this is from.
It's a grivet.
It's a grivet, right? It's kind of like a
vervet.
A vervet, but
not.
Okay. So this supports infection with SHFV and PRRSV. Okay. So what they do is they do a genome-wide CRISPR knockout to say if we knock out a gene, is it a receptor? So they look for cells resistant to infection, and they find 2 genes. One is the Fc gamma receptor and transporter gene, and that encodes the neonatal Fc receptor, and then beta-2 microglobulin, which is needed for expression of FcRn. So it looks like FcRn is important because, again, you do CRISPR to knock out random genes, you infect with your virus, you take cells that are resistant to infection, and then you ask what gene is disrupted by the CRISPR, which you can do really, really easily.
And CD163 shows up as a sort of an internal positive control.
That's right,
Yes.
which is good. Because if someone came to Rich and said, "Boss, I got this result," but he'll say, "Well, where's those CD163?" And
Yeah. That's right.
go back,
That's
do it
exactly
again.
what I'd say.
Yes. Well, and this is sort of cool because FcRn is known among immunologists as a thing that helps antibodies cross different cell or tissue layers in the body, particularly that helps antibodies cross the placenta by binding to the Fc portion of the antibody when it's in the blood and then helping that transcytose across the placenta. You can also see it involved in antibodies transcytosing from the blood into breast milk.
Right.
By crossing cell layers. And, that, immediately makes me think, that might have something to do with those, with the pregnancy issues in the animals that Vincent mentioned, that these viruses are known to have.
But it also, correct me if I'm wrong, but that means it's designed to bind stuff and internalize it.
Yep. Bind stuff, internalize it, and not have it get degraded.
Yeah. Okay. All
So
good things if you're a virus.
Yes.
So they take a cell, they take MA104, the cell line, they knock out this gene. They show that the virus cannot infect. Echovirus 30, a different virus, is a picornavirus, also supposedly binds to FcRn, doesn't infect those cells. SARS-CoV-2 does infect the cells because it doesn't bind to FcRn. And then they take a human cell line and they have a cell line which is lacking CD163 and it has CD163 but lacks FcRn, completely resistant to infection. So it's interesting, right? So obviously CD163 alone is not enough. You need both.
And they assess all these things, not only by doing growth curves from virus, but they have these beautiful pictures of cytopathic effect. Mm-hmm. That is the photomicrographs of the virus making the cells round up and croak.
And then
old-time virology.
When they put Grivet FcRn into this cell line that lacks FcRn, they can overproduce it. They get a lot of virus. It makes it hypersusceptible to infection. So does this say, Dixon, that this is needed for entry? I'm sorry to put you on the spot.
Dixon, you're muted.
That's okay. I know you know the answer to this.
I'm delaying my answer because you caught me in the middle of looking up what a Grivet was. I know what a vervet is, so I've seen vervet monkeys, but I don't know what a Grivet.
Nan can tell you.
This is not well. I'm tempted to say yes, but I think that would be the wrong answer.
No, it's not. So
good.
What they do is they take these MA104 cells that lack FcRn, and they put the viral genome into the cell, bypassing the receptor. And they show even without FcRn, the virus can reproduce in the cell. You can get infectious virus. So that's a really nice indicator that the FcRn is needed for entry. That experiment goes way back to the 50s, to John Holland. He did that with poliovirus. So mouse cells, if you throw poliovirus onto mouse cells, it doesn't infect them. But if you take polio RNA and put it into mouse cells, it grows beautifully. So he said, there must be a receptor. Oh, wait. So then they make monoclonal antibodies against, they screen a panel of anti-FcRn monoclonals, and they find one that blocks infection. So that could be therapeutically useful, right? If you have an outbreak on a farm, maybe you could treat it with, but then the vet will come and say, you should have bought those FcRn null pigs, no, CD163 null pigs, then you wouldn't have this problem. Okay. Then they do some interesting experiments to take this further. They put FcRn, CD163 or both into wild-type MA104s, which have very little of those proteins, and they infect. when you put a lot of CD163 in, it doesn't matter so much. It's FcRn that really makes the difference. And they do plaque assays, Rich.
I saw that.
Very nice plaques.
Yep.
And in fact, you get more plaques when you have more FcRn. And they say, well, we probably have been underestimating the titers of our stocks or our virus stocks because the cells have given us fewer plaques unless you have a lot of FcRn in them. I think that's very cool. Larger and well-defined plaques. So the cells don't make more virus. It's just the indicator that you do the plaque assay on is more sensitive.
Right.
I like that. So they ask, could you dispense with CD163 entirely? So
Don't ask me.
And I wouldn't. So if you don't have CD163, but you do have FcRn, they can't support infection. You do need to have some CD163 present.
Which is good news for those knockout pigs.
Exactly.
Oh, yeah.
Exactly. Do you think they taste different, Brianne?
I don't know.
They are GMOs.
They would be.
That's right.
So Viro is another cell line, right?
Mm-hmm.
Which is from Viro.
Viro is, we'll say, from Vervet.
Vervet, sorry.
Yeah.
And the MA104 is from Grivet.
Yes, although, again, if we go into the weeds of differences between Vervet and Grivet, it does get very weedy.
Okay.
If you look at the pictures of Vervets and Grivets, you can't tell them apart. So
Viros,
really, they look the same.
Viros are not susceptible to infection with these viruses, these artery viruses. And so these Viros don't have FcRn and CD163. It's very interesting. So they put these two genes in, FcRn, CD163, and if you put a FcRn gene alone in, it's not enough to give infection. But if you add CD163 alone, you get a little bit. But if you put both, you get nice infection. So you need both, even in Viro cells. But they're putting Grivet into Viro.
Yeah. Again, there's a lot of weeds here.
But I wonder how different they are, right?
They're not super. So the problem is that there was one species, and then people have since realized that there were subspecies.
Mm-hmm.
And so there originally was one species, which was Grivet, and now they've been sort of divided.
Mm-hmm.
I look forward to Jens's letter.
I think we have one in the queue.
Yeah.
Okay, so they wanted to know if CD163 seems to be a species barrier, and they want to know if FcRn is the same. So they get FcRn DNA from Grivet, pig, human, mouse, and horse. I love these zoo experiments.
Mm.
They put them in cells, MA104s that lack FcRn, and then they infect, and they find SHFV can only get in cells with Grivet in human, CD163, PRRSV can bind them all. One CD163 for them all. To bind them all. Isn't that from the Lord of the Rings?
It is from the Lord of the Rings.
One ortholog to bind them all. And in horses, didn't care. Horses don't care.
No.
Which is a typical horse thing. They don't give.
And they've left the barn anyway, so what there's
Why the long face?
Exactly. We've got a Scotch named after you.
Did you hear the joke? A priest and a rabbit walked into a bar, and the bartender said to the rabbit, "What are you doing here?" And the rabbit said, "I think I'm a spell check error."
I like that. Oh, that's
Yeah.
good. That's very
A little
good.
corny.
That's very, very good.
Okay, so FcRn. So a similar pattern for FcRn. So SHFV can bind Grivet, human, and horse, but not mouse or pig. So it's a species barrier, just like CD163. Let's see. Oh, this is a cool experiment. So SHFV apparently is the only one of these viruses that can be grown in immortalized cell lines. So they said, well, maybe we could take FcRn and CD163 and make MA104s susceptible to different. So they have all these other viruses with KRCV and PJBV. And so for KRCV, it will grow in those cells, but not PJBV.
Vincent, when you were a kid,
Uh-huh.
did you have a propensity towards alphabet soup? You just rattled that off like it was second nature.
As an immunologist, I definitely did.
Yeah, the immunologists are the kings of that. Right?
Well, you did pretty well.
Well, you would like to know what they are, right? Maybe let me go back and tell you.
I would love to know, what is the difference between all those species CD163? What is the difference?
Oh, well, that's a good question. Here we go. KRCV is Kibale red colobus virus 1. Oh.
Colobus.
Mm-hmm.
Colobus. Where's Kibale?
I don't know.
Oh.
Must be Central Africa.
Yeah,
Oh, maybe Uganda?
Yes, Uganda.
The colobus monkeys are...
And then PBJV is Pebja virus, caused an outbreak at a primate facility.
Pebja virus?
Pebja, PEBJAH. Anyway, so they just... So the MA104 cell line works for one, but not the other. But anyway, this is what you can do with viruses and receptors. And they have now this second receptor, FcRn, that's needed along with CD153. So they think, maybe the virus binds CD163 and FcRn takes it in because, as you said, Brianne, this molecule is involved in bringing things into cells, right?
Mm-hmm.
Vincent, I just thought of a great board game. It's called Viruses and Receptors.
How would it work?
Well, actually, they're magnetized or not, depending on whether it's the right receptor or not. And you shake it all up, and only the ones that should stick do stick.
There's a problem. There's a problem with board games.
I guess.
Maybe it's a bad name, right? Because it sounds like you're bored.
It might be that's true too. Board stiff. That's right.
So here we have a little. This is very interesting. In normal physiology, IgG and albumin bind to FcRn. They are endocytosed and then released before they get degraded. And so they say maybe viruses are doing this too. They're getting into the cell and being released and trafficking into a degrading pathway, which, the endocytic pathway can bring things to degradation, but you don't want to degrade IgG. You would like to release it beforehand. So that's the paper. What else? Plaque assays are very cool. Oh, so they say this is important for virologists because the MA104, which has been the gold standard for decades for quantifying infectivity, inaccurately underquantifies infectious arterivirus titers.
Interesting.
So now we're going to fix that. And maybe they could use some monoclonals to treat the infected animals. So nice job.
So I'm astounded at how many different experiments they ran to put a circle around the problem so that they could contain it. Did they leave any out that you would like them to run?
Let's see.
Oh, great wizard of the viruses.
No, I'm not a great wizard, but they did a lot
well, you are compared to me.
They did many of them. I mean, I have to tell you, when we published the polio receptor in 89, it had one of these experiments. It had just one.
Well, that's
oh my gosh.
I was asking, when do you stop doing experiments and publish?
Unfortunately, you sit down with your group and you say, we think this is good. Then you submit it, and then the frickin' reviewers tell you to do more. Right?
Right. Yeah, they do. They have the chutzpah.
And then you have this paragraph which is kind of tacked on at the end, which is obviously reviewer 3's experiment, which you didn't care so much about.
Right.
But I think a paper should be reviewed on what is submitted.
Correct.
Don't ask for more experiments.
I totally agree with you.
And if it doesn't work, then say we're not taking it. That's what
I mean,
Eli does.
this is a good story, though, right?
I like this very much. I think it's a nice story. By the way, the first author is Teresa Shaw. I think there's just one first author. And then two last authors, Cody Warren and Adam Bailey. I like it very much.
It was actually submitted in March of 2024 and accepted in July, so not too long.
That's not bad.
Not too long.
Okay. Then we have a second paper, which is about plants. I haven't done a plant paper in so long. Cell host and microbe. Protein arginine methyltransferase 6 mediates antiviral immunity in plants.
Can you tell me, in simple terms, why they inserted the word protein? Because any enzyme is a protein. So what's that word protein doing there? It's confusing.
The reason is that this is sort of a general family of proteins, and this PRMT6 is known in other, like that's its name, and it's been known in other contexts. So somebody in the past named it that, and it's stuck.
Oh.
So it's historical.
That was a disappointing explanation.
I'm sorry.
Happens.
I thought it had a deeper meaning than that.
No.
Because maybe not all the arginine methyltransferase 6s are proteins. Maybe there's...
Well, maybe you could also say, well, maybe this doesn't act on free arginine. Maybe it only acts on arginine that's in a protein already.
I see.
Yeah.
I see.
So it's not the methyltransferase that the protein is referring to.
I think it's referring to the
methyltransferase is the target.
Yeah.
The protein is the target of the methyltransferase.
The methyltransferase
that makes sense.
Is modifying arginines in proteins.
Right.
Okay. I hear you. Right. Now I hear you.
All right. So
you could ask where the hell else are you going to find arginine?
Exactly.
Yes.
We won't go there.
But as Brienne said, someone just gave it the name, right? It was probably a long time ago when we weren't pedantic about names.
No.
Two first authors, Shang Zhu and Ayaz Ahmad, and two last authors, Feng Li and Xiaofeng Cao from Huazhong Agricultural University, Chinese Academy of Sciences, and Hubei Hongshan Laboratory, of course, all in China.
I like that it's called the National Key Laboratory for Germplasm Innovation.
Wow.
That's amazing. Huh?
Yeah.
And utilization of horticultural crops. Don't leave them out.
No.
Okay. Plants get infected with viruses, right? Just like everything else.
Plants don't have antibodies or T cells.
They don't have antibodies or T cells. They have macrophages. They don't have microglia. They don't have brains.
None of that stuff.
No.
But in defense is RNA silencing. It's nucleic acid-based antiviral defenses.
Well, they also have some defenses that are similar to some of our innate immune receptors, the NLRs.
Okay.
Which they talk about a little bit here, because there are some similarities to those NLR proteins here.
So the way this works is viruses are reproducing, and double-stranded, these are RNA viruses here, but it could work for DNA as well. RNA viruses are reproducing, getting double-stranded RNAs made. They are chopped up into 21 and 22 nucleotide RNAs that are called small interfering RNAs. There's enzymes in the plants that do that. They're called dicer, which is a beautiful name, right? And then they're methylated, and one of the strands of the double-stranded RNA is incorporated into a complex called Argonaut, which is also wonderful. And it makes what's called an RNA-induced silencing complex, because now that little RNA, that 21, 22 nucleotide RNA, is going to find a viral RNA, and it's going to bind to it and chop it up because it's got a nuclease, the RNA-induced silencing complex. So that's how RNA silencing works. And these can be amplified. These little RNAs can actually be amplified in plant cells by polymerases, and they can move from cell to cell via these things called the plasmodesmata and phloem, right?
That's right.
So if this is a wonderful system, it's very efficient, and if viruses could not antagonize it, they wouldn't exist, right? So they have what we call VSRs, viral suppressors of RNA silencing. Okay, and they inhibit multiple steps in this siRNA pathway that I briefly discussed with you. There are all kinds of steps, right? Like dicing, they sequester, they degrade proteins, argonaut gets degraded, all kinds of different mechanisms, all right? And one virus that is a model system for understanding VSR is called tomato bush stunt. You know, in the old days, this used to be tomato bushy. Did they change the name?
Don't know.
Because
Don't know.
throughout the paper, it's tomato bush. That makes sense. It's a tomato bush. Okay.
Yeah.
It's tomato bush stunt
and the
virus.
virus stunts its growth.
And it encodes a protein called P19, which is a viral suppressor of RNA silencing. And what that does, it promotes systemic movement by binding to the VS RNA complex. And what's really cool about this is that they've done structural studies on P19. It has a concave face that binds on one side of the siRNA helices, and there are two tryptophans that cap both ends of the helices, so that it knows how big of an siRNA to bind to. It's just really nice, and that's going to be important, as you're going to see.
And if people want to understand this better, I did really enjoy the graphical abstract.
Yeah, I'm sitting here looking at that. It's very good. It took me a while until I could get a little bit into the paper and sort out who all the players were. But the graphical abstract is actually quite good.
So the P19 has a homodimeric protein that will bind to this siRNA and prevent it from working, basically. Okay, but there are other strategies as well. As Brianne said, there's this NLR nucleotide binding leucine-rich repeat-containing immune receptor mediated immunity, and they talk about it here a lot. We don't need to go into that, but protein, arginine, methyltransferases may play a role, and that's what this paper is about. It hasn't been previously shown to be regulating immunity. And they find a new role for PRMT6. Let's shorten it, PRMT6.
So what's really interesting here is that PRMTs, particularly PRMT6, are important in other types of immunity.
Mm-hmm.
And so if you do a PubMed search, you'll find all sorts of things about PRMT6 and immune responses, but no one has actually looked for it in plants before this paper to see if it does anything with immunity in plants.
Right. So they have. So what they do, a series of experiments, they use Arabidopsis, which is a model. It's a weed, but they use it as a model organism. It's been elevated to model status.
But it's a flowering. It's an angiosperm.
Actually, I think using the word weed isn't fair because everything.
It's not fair at all.
Right.
It's the
It's
Drosophila of plant biology.
It's a plant. What the heck is the weed business?
Yeah, exactly. A weed is really a subjective term.
Exactly.
That's just a plant that's growing somewhere where you would rather it didn't grow.
If you're a farmer,
that's just you, okay?
If you're a naturalist.
Like your garden, Rich, right?
Yeah.
That's right.
So, previously it's been shown that PRMT6 is induced by virus infections in plants. And what they found is that the PRMT6 of Arabidopsis inhibits the VSR activity of P19. Remember, P19 can antagonize siRNA, and so PRMT6 inhibits that. So maybe this is a little hard. All right, so the plant is defending against the virus by siRNA. The virus has P19, which is countering that, and now the plant is going back with by presumably putting methylating arginines on P19, but we're going to find that out.
These sorts of things do get very confusing because it's a sequence of negatives. Okay,
That's right.
That leads you wondering where you are when it's all over.
So basically, we are learning that PRMT6 is stopping the evasion mechanism. So the virus is trying to evade and sneak around, and this is basically shining a light on that evasion, so evasion no longer happens.
So what if you want to know if PRMT6 is important, you knock it out, right? So they can do that in plants. They knock out with CRISPR-Cas, and they have 3 knockout cell lines for PRMT6, and then they infect them, and they look to see how they react to virus infection. And they have severe symptoms, moderate symptoms, and mild or asymptomatic symptoms. My favorite is the dwarf and leaf crinkle phenotype. But basically, if you knock out PRMT6, you reduce immunity because the tomatoes are getting sick now, right? You get
Well, no, it's the
symptoms.
Arabidopsis is getting sick now.
The Arabidopsis is getting sick.
This all sounds like fantasy world, but the indoor farmer now, the industry that has come up because of the horticultural interest in tomatoes, is having fits with these viruses.
Yeah.
Fits.
Oh yeah.
It's dooming a lot of indoor farming, basically.
It's I'm just looking over this again. It's not precisely clear how they focused in on PRMT6. My guess is that there's more to that story than is related here, except that, I mean, the only thing they really say is that PRMT6 is induced by the virus infection. Okay, so that makes it a suspect for something going on.
But Brian, these are tomatoes, actually.
Oh, is this tomato or is this Arabidopsis?
Yeah, well, the inhibition is done in Arabidopsis initially, and then they knock out in tomatoes.
Oh, okay.
Yeah, because like I said before, but I think I was overshadowed by the chatter. Arabidopsis is the Drosophila of the plant world.
I heard you.
Oh, you did? Okay, fine.
Yeah, it's
Just
good.
wanted to emphasize that.
So the nice thing about this paper is they make a conclusion at the end of every section. These results showed that mutations in PRMT6 reduce immunity to TBSV in tomatoes, resulting in enhanced viral systems. Okay, so then they ask if you overproduce PRMT6, what does that do to immunity? So they make transgenic tomato lines that overproduce, and yes, if you overproduce, you get enhanced immunity against TBSV in tomatoes, so you get less symptoms. Pretty straightforward. So what's the mechanism? This protein PRMT6 can enhance immunity against TBSV. What's the mechanism? To do this, they use bimolecular fluorescence complementation, which is so cool.
Get out.
Yeah.
Shut the door.
So they have a fluorescent protein, and they cut it in 2 parts, and they put 1 part on one protein and 1 part on another. And if those proteins interact, they're going to restore the fluorescence. Isn't that cool?
That's a great trick. Great trick.
I love it. So they have a couple of TBSV encoding genes, including P19. They put it the half of the thing there, the fluorescent protein there, and then they put the other half of the fluorescent protein in PRMT6, and then they show that P19 binds PRMT6 because it glows. They confirm it by co-immunoprecipitation, and they do by tagging the protein. So apparently the proteins physically interact. Not surprising, right? Then they have a cool assay for viral suppressor of RNAi. So they have cells making green fluorescent protein, and they produce in those a microRNA against GFP, which is going to reduce the fluorescence, and then P19 is going to reverse that inhibition of fluorescence. And if they add PRMT6, fluorescence goes back down. So it rescues the siRNA inhibition of GFP. So they're using a model fluorescent protein to measure siRNA, just makes it easier.
So that basically focuses your attention now on the mechanism being the binding or not of the microRNA. Right. The PRMT6 makes it so that P19 is no longer binding the microRNA. right?
Right.
Right. So the way that P19 works is to bind the RNA, the siRNA duplex, right? And so they ask next, does PRMT6 reduce the affinity of P19 for the siRNAs? So they do RNA immunoprecipitations, and it does. If you have PRMT6 present, P19 does not bind as well to the siRNA duplexes. And
One thing I
furthermore,
like here is that these assays, now we're in plants here. They have this wonderful picture of a leaf that has basically the whole experiment on it, where these various expression plasmids have been added to patches in the leaf. And you can look under appropriate lighting and see whether or not there's fluorescence. Very visual, very cool.
yeah, visual, it's cool. So they further show that, in fact, what PRMT6 is doing is preventing dimerization of P19. So you need two P19s to come together to bind to that siRNA. And so apparently they have an assay for dimerization. They show that PRMT6 is preventing that. And so since PRMT6 is a methyltransferase specific for arginines, you would think that maybe arginine is the target, right? So
Hence the title.
They do in vitro methylation, and they can detect the methylation of arginine by mass spectrometry. And what they do is they change arginines in the P19 and show that, in fact, I think two of them are substrates for methylation by PRMT6. And in fact, they can show that those arginines that are methylated, if you change them to something else, the protein doesn't dimerize and it doesn't have any activity. Very cool. They change each of these arginines to alanines, and they show that if you're an alanine, you can't be methylated, and so the protein loses activity. No, you can't antagonize the activity of the protein, right?
Right.
PRMT6 inhibits the SI binding activity of P19 by methylating their two amino acids that are important and regulating dimerization. Now, this is cool. Well, this is in plants in the lab, but what about in the real world? In the real world. So now we have two arginine residues that are important. What if you look in plants, you see any variations or single nucleotide polymorphisms? So they have a whole database of sequences from plants, because you can imagine that this is a big deal, right? Crop breeders are always sequencing their plants. So they
want
found
to grow
one.
more tomatoes so I can eat tomato sauce.
You betcha.
Tomatoes are wonderful, right?
Exactly.
Tomatoes are wonderful. And they find a SNP, a certain SNP that seems to be it's at one of these arginines, so that's a candidate variant, and they go ahead and show that in fact it does make a difference. And then they look in the population to see if the expression level of PRMT6 is associated with resistance. So there's some crops that are more resistant than others, right? So they look for this SNP that they've identified, and one base at that position, the plants have mild symptoms, and the other base they get more severe symptoms. So the expression level of PRMT6, and that's associated with expression levels of PRMT6. I didn't mention that before, but the plants that have milder symptoms have higher levels of PRMT6 than the plants that have more severe symptoms.
So plants are different from animals in another way, and that is that they're filled with ploidy, multiple copies of the same genome. And like orchids, for instance, have presumably the world's largest genome because it's got like 8 ploidy or 12 ploidy or something like that. So how does this work out? I mean, do they look at each of the ploids to see whether they're all this way or just some of them are that way?
It's a good
Could
point,
that be a
Dixon.
defense mechanism? Could
It's
ploidy be a mechanism for avoiding some of these ill effects of a single gene change? Ah, that's okay. I got 10 more. It doesn't
I
matter.
think that's a good point. It could.
Just keep going.
Certainly could, yeah.
So that looks like a way of developing not immunity, but avoidance.
They may have it in the methods I didn't see, but in the text they don't mention that. But yeah, you're talking about gene dosage, basically.
Yeah. That's right. That's right.
This last experiment with the natural variants of the plants
Right.
was really cool and for me critical because leading up to this, some of the effects that they were talking about in their in vitro experiments, I thought were pretty modest. Okay. Although consistent with Dixon's highlighting the previous paper, they, do it from so many different angles. They encircle the target that it becomes believable. But this, with these variants, is completely consistent with everything that's gone before. They're natural variants. You didn't do anything. And the effects are pretty dramatic.
Yeah.
The difference between the resistant and the susceptible plants in terms of both the plant size and the amount of virus it's made is striking. So I thought that was a really cool experiment.
Right.
So I didn't really explain it well. Let me redo it very briefly. So they have SNPs, single nucleotide polymorphisms, in PRMT6 that correlate with expression of the protein. So they select plants that have high and low expression of PRMT6 and they infect them. And they show when you have high amounts of PRMT6, you have fewer symptoms, right? So expression level is positively associated with resistance in natural populations. So this is informative because then you could breed crops to be more resistant, right?
Right.
So this is cool. That's the paper. PRMT6 is involved in antiviral immunity, counters S19, which in turn counters plant siRNA immunity.
And also kind of cool to know that these methyltransferases that are acting on arginines are acting in immunity across kingdoms.
Yeah, I didn't realize that human PRMT6 restricts HIV.
It restricts a bunch of viruses.
That's very cool.
So when they talk about doing this screen to look for a SNP associated with PRMT expression, they say the expression data of PRMT6 in pericap were extracted from 401 tomato accessions and range from 4.91 to 28.23. So this is all in silico, right? This is pre-existing data. Is probably RNA expression or something like that.
Yeah.
And these are all out there in the database so that you can just go screen for who has data on different genome expression of this protein from different tomato genomes, and your data are all right there for you to just analyze.
Rich, I would just call that cherry picking. It's great that you can do that, though.
Yeah.
That means that there's a hell of a lot more interest in plants than there is in animals, because I think without plants, you don't have animals. Period. That's one way to look at that. And
it's interesting. I didn't think of that.
Yeah.
So they mentioned that other viruses infect tomatoes, and they have conserved VSRs that are probably targets of PRMT. There's also PRMT5 that restricts hepatitis E virus, and herpes simplex virus. Wow, this I did not know. That's very cool.
There is a statement, however,
Huh.
in one of their paragraphs, which I am struck by, and that it says, however, whether PRMTs, no number, play a role in plant immunity remains unknown. Now, did they solve that for PRMT6?
Where'd you see that, Dixon?
It's on page, it says XS3 organization. It's just above that statement. I don't have a page number for that.
Is it towards the end of the paper, the beginning, or the middle?
No, it's near the end. It's the beginning of the section snippets, just above it. It's the second paragraph before that. It says, however, whether PRMTs play a role in plant immunity remains unknown. Would they say that before they reveal the results?
I remember that in the intro. Yeah, that's in the intro.
It is in the intro.
Yep. It's the second to last paragraph of the intro.
That's right. So they've answered that through all of these experiments.
Yeah.
Okay. It should
All
say until now.
So I hope we did justice to this plant virus paper. I know there's some plant virus people listening, but if we made mistakes, let us know.
Right.
Let's do a couple of emails here. Dixon.
Yes. Megan writes, saw this floating around and wondering if either Daniel or the TWiV folk could address this as part of the next H5N1 coverage. My understand is this is pretty far across the state from where the currently affected farms are, but I don't know exactly what goes on into the water and the wastewater there. Of note, I work for a medical system on Rhode Island, and we received information this week that if someone works on a dairy farm or has been exposed to dairy farms or raw milk and presents with influenza-like illness or conjunctivitis, you should isolate them. And then they give a link for the wastewater reference.
Huh.
Megan also writes, quotes, to lend each other a hand when we're falling, perhaps that's the only work that matters in the end. And that's a quote from Frederick Buechner. I'm unfamiliar with that.
So I think this is detection of H5 influenza virus in this Coralville, Iowa wastewater, right?
Right.
Mm-hmm.
On just one sampling.
Yes.
In
and she says
early July.
It's not near where the farms are affected.
Yeah, well.
But, people can move around, right?
If you just take one sample and you find it, that's
I
probably going to be a lot of other places too.
Birds can move around too.
Yeah, birds do. That's right. So,
gee,
I think one is just, it doesn't mean a lot, right? But if you saw multiple now, then it would mean that there are infected animals in the vicinity.
Right.
So, is this updated? I think this is the 8/12, which is today. Yeah, so there's not been anything since this one sample, which is July 10th. So I would say it's nothing. I mean, it's something, but it's not an outbreak, right?
It means it's hanging around somewhere.
Why did I put this next one in?
Because your name is mentioned.
No, I don't like this. Let me see what this is. Oh, okay. Let me read this because I know what's going on here. This is Daniel. I've been arguing with many of my friends who have advanced degrees about the lab leak hypothesis. Well, that's your mistake right there. Recently, one of them sent me this as, quote, proof that the lab leak hypothesis is correct. I thought it was a lot of gobbledygook, but thought you and your team might be interested in what's out there. Okay, so he sends a link to a Substack where you can write anything you want. And basically what they have done here is to use Bayesian statistics to say whether the virus SARS-CoV-2 came from a lab or from nature. Okay, and many people have done this, and some people it says nature, and some people says it finds lab. You know, it all depends on what you put into
Yeah.
the analysis,
Yeah.
right?
Mm-hmm.
Yeah. The way I saw this is it's, some sophisticated looking probability analysis. Okay. Trying
Yeah.
to figure the probability that this would be the case. But, it's nuts because
Nature doesn't work.
it all depends on what you decide the input probabilities of this or that are.
Yeah, isn't
So-
this kind of a garbage in, garbage out problem?
Exactly.
Yeah, absolutely. Very well
You know,
said.
66 million years ago, a meteor hit this planet and wiped out 75% of the life on Earth. Predict that. Put that in your telescope and smoke it.
So that's what we think that is, Daniel. There's nothing
Yeah,
to
he
it.
called it gobbledygook.
Yeah.
Right on.
Gobbledygook.
Brianne?
Fernando writes, Hi TWiV hosts. I really appreciated Alan's pick, being a longtime fan of Tumane Di Bite, who I had the delight of hearing him at several memorable live shows. He was such a warm, wise stage presence, even though he suffered throughout his life from the long-term effects of childhood poliomyelitis, which forced him to use a crutch and to move with difficulty on stage. He died of kidney failure at 58, but he had been ailing for several years, causing him to cancel his frequent trips to the USA where he had family. Thank you for the depth and breadth of your interests, even the weather, which makes me feel like I'm hanging out with y'all.
There you go. Another good route for the weather.
Who's next? Did
Me.
Rich go?
Which
Me.
goes next?
Go ahead.
Peggy writes, Dear TWiV, I live in the UK. Totally agree with Rich that roundabouts should be used more in the U.S. I recently did a road trip from L.A. to San Francisco and frequently noted how roundabouts can be better in most intersections. This, I have to say, a little interjection here, occupies a significant fraction of the conversation that I have in the car with my wife when we're going somewhere. A roundabout would clean this up, right? Also, the stop signs in small street intersections drove me crazy. Here we use a yield or give way road marking to indicate who has priority. This means that if the road is empty, you can slow down a bit to make sure it's safe before accelerating again, not stopping at all. In the U.S., I don't know where this name came from, but that's what I grew up understanding as a Hollywood stop. Okay, where you pull up to the stop sign and kind of have a look around and not stop completely, but look safe. You'd go on.
Well, I feel like you must not drive frequently around New England and Boston, as you're referring to these as roundabouts instead of rotaries.
Yeah. Well,
Which there are many.
think they're roundabouts.
Yes,
Okay.
I think it's a big distinction of whether they're roundabouts, rotaries, or traffic circles.
Right.
Okay, Dixon, we're back to you.
Felicia writes, I would like to suggest that you consider having Dr. Sabrina Schultz, a biological anthropologist, on your show. And Felicia gives a reference to how to get in touch with her. She recently authored the book, The Human Disease, and it was produced by MIT Press. She was the lead curator of the exhibition Outbreak, Epidemics in a Connected World, at the Smithsonian National Museum of Natural History in Washington, D.C., and scientific commissioner of the exhibition Epidemics Apprendre, I can't do that, sans du vent, I guess it is the Musée des Confluences in Lyon, France. I've been to Lyon.
Epidémie, prendre son du vivant.
Merci. Felicia was on TWiV. She curated the Outbreak exhibit at the Smithsonian. We recorded the episode in the Outbreak space. So we did that. And you want to do it again?
That was such a good exhibition.
Ed Niles was on that TWiV as well, because
he was. That's right.
Because he was a docent at the Smithsonian, and helped guide people around that exhibit.
Yep, it's a great exhibit. Cyril writes, hi TWiV, goes without saying I love the podcast. 2 items. I have this ongoing discussion with a mod on Reddit. They keep referring to Vincent as Vince. I never heard anyone on the podcast or anyone anywhere refer to Vincent as Vince, but this mod said he called Vincent Vince and said Vincent was cool with it. Does Vincent go by Vincent or Vince? I don't like Vince.
Just like
Yeah, the reason you don't
Or
hear
Vinny.
people, the reason you don't hear people on the podcast refer to Vincent as Vince is that we know better.
I just don't
That's
like
true.
it. I mean,
we want to live.
I prefer the Vincent. Yeah,
You do.
I
And,
do.
and now you don't even like Vinny.
Vinny is okay. Yeah, if you know me, Vinny is okay.
Vinny, really? Oh, I've been
I don't mind
for Vinny.
My cousin
Okay.
Vinny. Yeah.
I love the movie.
Okay. Next point. On your list of guests on the podcast, I do not see Shane Crotty listed. He was on 3 episodes after I did a search of your website. That's right. At some point, I just fell behind in listing all the guests, and I'm sorry. Maybe we'll get someone to do it.
Well,
one day.
but you know what? It turns out that Google searches of TWiV plus whatever you want to know work just great.
That's true. They do work really well. Okay. Yeah, we got one more left. Let's go, Brienne. It's your turn, right?
Anthony writes, in quotes, that said, while coronaviruses do infect some bats, the SARS-CoV-2 virus has never been found in any bats. And then Anthony continues, an expert on bats recently sent me this. Of course, it's at the same time true and not true. Considering the vast area of rural China and that neither the government of China and some facets of the U.S. have not encouraged such a search, the lack of data is not hard to understand. Plus, given the nature of quasi-species, or RNA swarm, whatever the mot du jour is, SARS-CoV-2 will most likely never will be found. What will be found are various related viruses that rose from the same sources. By the way, listening to people who should know, I am surprised when they wonder why there's talk of a last common ancestor with rinderpest and measles. Whether the viruses diverged 500 years ago or 1500 years ago, or even if it had been 50, the fork in the road gives 2 different travelers. Most educated people reading the bat expert's statement could not be faulted for concluding that SARS-CoV-2 was fabricated. Perhaps you could consider doing a virology quick start for policymakers, pointing out the vast geography involved in the development of influenza and the bat coronaviruses, plus the nature of quasi-species, the huge number of progeny, and quick generation time have as much to do with Mendel's peas as a trip to another galaxy has to do with a ride to the supermarket.
So I excerpted a paragraph from our rebuttal of the Chan Times opinion piece because she also wrote this, bats in other parts of China have not been found to carry viruses that are as closely related to SARS-CoV-2. And so the response is, the closest relative to SARS-CoV-2 isolated in China is RATG13, which exists only as a genome sequence, not infectious virus. It was collected in the Tongguan Cave in southern Yunnan province, quite distant, 1,800 kilometers from Wuhan. It is 1,200 bases different from SARS-CoV-2, 96.2% similarity, hence cannot have been the progenitor. Other similarly related viruses have been discovered in Laos and from Malayan pangolins. The RBD of these viruses is nearly identical to that of CoV-2. These viruses have been circulating for many years. So this is used as an argument that it's man-made because we don't have anything close to it, but it's just a specious argument, right? Okay. Time for some picks of the week. Brienne, what do you have for us?
So I, as many people have, been watching the Olympics a lot of late, and I really enjoyed the story of a particular Olympian named Kate Douglas, who is a U.S. swimmer. She won 4 medals at the most recent Olympics. She is also currently a graduate student in statistics, and has actually published a paper on statistics data analysis and swimming, where she and her colleagues basically were able to digitally look at swimming positions of different swimmers and determine things that were related to swimming times. She was actually able to identify an issue with her head position in her swimming and changed it. And then in this Olympics, won 2 golds and 2 silvers. And dramatically improved her times. And so she is both a recent Olympian, but now also a statistics graduate student who's doing some really cool work in statistics. And she published this paper this year called Swimming in Data, where you can see images of her swimming compared to some other people, and then the data that they get from their digital twins and how they've analyzed it. And so I thought that was a pretty cool story of how this woman has sort of used her statistics knowledge to improve her swimming.
Cool.
How wonderful.
It's all science, right?
Yeah.
That is wonderful.
I didn't watch much of the Olympics. I watched a few swimming events, and it's interesting you should bring this up because the underwater shots of the swimming are terrific.
Mm-hmm.
And I remember watching one of these races and thinking to myself that every micro portion of the moves in their strokes is analyzed and computed and practiced,
you can see
and
it
they
here.
figure
Yes.
it out. I noticed one race in particular. I think it was a butterfly race, where one of the swimmers actually, after diving in, coasted for an extra long period of time. It seemed like forever, where the others had already taken off and stroking. And I was wondering to myself, is that even legal? Okay.
No,
But
maybe not.
and thinking to myself, that is a very calculated move.
Absolutely.
But they're all
Yeah.
calculated moves. This is really interesting.
Well, there's less friction under the water than there is at the surface.
Yeah, well, that's what I was thinking. I mean, butterfly is a really interesting stroke because on the one hand,
It is.
it's
That's right.
beautiful, and I
Yes.
cannot do it. I cannot coordinate it.
I can't either.
It's impossible. Okay, and yet it's also really a violent stroke. It's beautiful.
Yeah.
But you're really thrashing at the surface.
You're right.
Yeah, generally when I try to butterfly, it kind of looks like I'm drowning.
Your rotator cuffs, beware.
Yeah, now we're going to get a bunch of mail from swimmers, which is good.
Okay. We haven't had a swimming arc.
No, we have not.
Dixon, what do you have for us?
I have a random list, not so random, of women of recent note. And I just started it with Kamala Harris, but then I said, oh, no, you can't leave out the Olympics because I've been watching them like crazy. So Katie Ledecky, I don't know what planet she's from, but she's not from this one because she's older than most of the other swimmers, and she's won this 4 times. The 10,000 meter swim is a boring swim, unless, of course, you're rooting for the one in front. And Ledecky was like an entire swimming pool ahead of everybody else for most of that race. Sydney McLaughlin-Leveron I picked because, of course, 400-meter hurdles, she broke the record, but she's also from New Jersey. So, that's a Jersey girl that I put in there. Then, of course, you had the 4x100 relay team who actually know how to handle a baton. Maybe they grew up learning how to throw the baton and then catching the baton, but the men have no clue whatsoever as to what to do with a baton, except to drop it or to not grab it when they should. And I'm being critical of the men's track team because that's the 4th time in a row of 4 different Olympics with 4 different teams that the baton played a huge role in their loss. And then finally, the women's basketball team. These are all professional basketball players. They beat France by 1 point, and they beat them in the last 2 seconds of the game because the French shooter had her foot on the 3-point line. If she had her foot behind it, she would have tied the game, and then it would have gone into overtime. At any rate, I was absolutely impressed with all of these women. And not only that this is the first time I was told by some announcer on the Olympics that this is the first time that the United States has 50% women and 50% men represented. The Olympics in general had 50% men and 50% women. And they said this is an incredible advance over the 1920s that had like 6% women.
Yes, and I don't know if you knew this, but for Team USA, 65% of our gold medals came from women.
Yes, I was going to get to that. We outmedaled the men like crazy, and not I think the speeds would have been different. You can't compare the racing of Noah. What was his last name?
Lyles.
Lyles, that's right. Who won the 100-meter dash in like incredible time. To the fastest woman who couldn't run that fast. Of course, built different musculature, et cetera, et cetera. But with the finesse sports, hands down, women had it. And then take a lesson from that.
So Sydney McLaughlin Levrone went to Union Catholic High School, which is about 5 minutes down the road from me.
Exactly. And she's a very self-contained and very humble person. And she could not believe how much she won that race by. But if you watch that race on TV, you'll see that it's just an amazing accomplishment for anybody to go through that. I'm, every time the Olympics come on, Rich, I watch every bit of it.
Rich, what do you have for us?
So a week or so ago, my pick of the week was Dillo Dirt.
Yep,
Okay.
I remember.
The stuff that's manufactured in Austin that is a combination of wastewater biosolids and compost that's collected around Austin and further composted and treated, etc., and is distributed as Dillo Dirt, and Dillo Dirt is like awesome. Okay, I've used it only as top dressing, but at any rate. So I told this story of my pick to my daughter, Sarah, who, as some listeners will know, is on the faculty at Texas State University in San Marcos, south of here. And she says, oh, on a related note, Texas State has a forensic anthropology research facility. Okay. And she sent me the link to it. The link that I've posted actually is a sub-page in a larger thing that's the Forensic Anthropology Center that has all kinds of stuff in it. But this describes the Forensic Anthropology Research Facility, which is a 26-acre outdoor human decomposition research laboratory. It's spatially the largest facility of its kind in the world, used by forensic science community to gain knowledge about human decomposition and developing methods for determining the post-mortem interval or time since death. And somewhere in here, it also points out that if you're going to do this kind of thing, you need to do it in different climates. Okay? So the data at this is specific for Texas and climates that are like Texas. So you can donate your body to this facility, and they will take your body out and lay it out in this outdoor in this 26-acre plot and watch you decompose and collect all the data from it and learn that. Okay? So I thought this was pretty cool. Reminds me of a book that was, I think, an Alan Dove pick from a long time ago called Stiff. Are you familiar with this book?
Yes.
Should have picked that along with this.
I remember that, yeah.
Stiff, very well written by I think, something like a forensic anthropologist with a sense of humor, okay, because it's written in an entertaining fashion that talks about all of the sorts of things that happen to human corpses after you die. All right, so there you go.
So there was a pick recently, I don't remember who, but I ended up buying Zen in the Art of Motorcycle Maintenance, so I'm reading it now.
Ah, okay.
You guys have read that, right?
I read it many, many years ago.
I have not read it, but I know of it.
It's good.
I skimmed it.
He skimmed it. Dixon. Shouldn't be skimming. All right. I have a pick from Science Magazine, an article by Catherine Offord. It's called Sea Lion Camera Crews Capture New Footage of Ocean Floor. So this is from a group down in Australia. They attached cameras, video cameras, to the backs of 8 Australian sea lions. And they're helping them map the seafloor habitat. It's like 110 meters down. And there's a video here which shows you it. And there's an interview with the people who did it. I think it's really cool that they're...
Yeah, the videos are great. Show the sea lions swimming through different habitats. And they make the point that, these sea lions are gathering data that would be hard to come by otherwise.
Exactly.
Right.
They're really good at what they do.
I'm waiting for the cetacean biologists to attach an infrared camera to a sperm whale.
Oh, how are you going to catch a sperm whale to do that?
They are actually friendlier than you think.
Yeah.
They don't like orcas very much, but I know a lot of people have recorded as being surrounded by it, and they can swim with them and everything else. And they were in the beginning very hesitant, but not now. But imagine an infrared camera on the back of a sperm whale that dives
Amazing.
to 3,000 feet,
Yeah.
and then they turn on the camera, and the life forms can't see the infrared. And all of a sudden, you've got giant squid.
That'd be cool.
That would be cool.
I would love to see that.
But this,
Well,
for these...
this was great, by the way. I watched it.
These
It
sea
was
lions, they had to take them out and put them on shore and attach to cameras,
Yeah. And the other thing they have now is they have lifelike robots of various animals
Mm-hmm.
that imitate some of the behavior of the
Oh,
live
I've seen
animals.
this, yes.
And it was on one of the Nova shows, and it was quite
Right, and they get
amusing.
great, they fool the creatures, and they get great pictures of
They
them.
do.
Yeah.
And they come over and they, like look at them and, okay, you're one of us. You can come along. A little slow, and he doesn't talk much. This is very interesting. This is great stuff. Great stuff.
All right. That is TWiV 1141. Show notes at microbe.tv/TWiV. You can send questions, comments, picks of the week. Yeah, you can send in your picks. It's called a listener pick. That'd be cool. Send them to TWiV at microbe.tv. And if you enjoy this work that we do here on these podcasts and all the others, we'd love to have your support. Microbe.tv/contribute. Dixon Despommiers at trichinella.org, thelivingriver.org. Thank you, Dixon.
Very cool episode. Very, very cool episode. Loved every minute of it.
Rich Condit is an emeritus professor, University of Florida, Gainesville. He is currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time. Great to see you all. Brianne, I feel like I haven't seen you for a while.
I know.
Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. It was great to be here. I learned a lot, and it was good to see you all, too.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.